Hello everyone, and welcome to the first episode of Data Disco. This is my attempt to learn bioinformatics in a fun way, not by textbooks, not by MOOCs or by Coursera courses, uh, just by getting a why and figuring out a how. It's not that simple, but yeah. So today's quest was given to me by ChatGPT, and I basically designed a prompt so that it gives me grounded quests to learn a topic and try and align it with industry-based skills, and not hallucinate and actually it be solvable, right? So if you're a regular on the channel, you know how I am at advocating AI as a co-helper of sorts to help you learn, to refine you, not replace you, right? So that is why I did that. So if I'll maybe post this quest in the description, you can go, like do it yourself. It would be much better, but this is not a tutorial. Let me make it clear. This is a walkthrough of sorts because what I have like always observed is that when you have a why to apply a knowledge, you learn much better. And when it is framed like an investigation, actually immersive, actually has real life application, I think it is much better. And especially a subject like bioinformatics, which is rooted in application, like uh, the theory is pretty boring, and like you don't really care a lot about theory. I'm not talking about the bio side of the theory, I'm talking about the informatics side of the theory, algorithms and all that. We have CSE people to solve that. So I don't mean that you're not supposed to learn that, but yeah, I am a firm believer in the project-based learning method. Like if you are building something, you're trying to solve a problem, even if it is already solved, but you're trying to solve it by yourself. Come to the solution by your entirely internal thinking or your own effort. You're going to learn much more than what you're going to learn with a textbook. So this is my attempt at learning, and this is something that I've sort of done to teach myself. This is the first time I'm using AI to generate a quest, and this has made my life much easier, I would say, and it reduces the friction between motivation, and then it just cuts off an extra step of framing the why. So, without further ado, let's just start. So, the first quest that was given to me was this thermophile enigma, and we're trying to find what makes thermophilus survive in uh, in a high temperature environments. Why did it take so much time to say that? So, it's not like I just used computer to just solve it. And there's a lot of I asked the prompt to not spoon feed me in any way, but like this is sort of my step by step sort of written thing. But the quest is this. Okay, it's just this much, and this is my extra output that I asked it for. But then you have to figure it out why you're doing this, how you, because this is not enough. I'll be honest with you. This is just 409 words, and the entire code is of about 400 lines. I don't know. So even if you're following along with the quest, it is not a problem as long as you are committing to it and you're committed to trying to find the answer. That is my entire, insight to you about this kind of approach. And if you want, you can try it by yourself. If you want a quest like this, you can ask me. Maybe we can solve it together. And one more thing, like it's very hard to explain things. I cannot explain the entire code because it took me 7 hours to do this entirely. I will try my best, however, to explain the entire process. I would love to, but I know that long form content doesn't really get, especially when all of our attention spans are dwindling due to short form content. Long form content is hard to make because you have to make edits. I am very edit averse. I don't like editing. Uh, so there's that. I would love to do that someday. I might even solve something in a couple of videos like series and sections. But I'm not the kind of guy to like, stop the flow state, because you don't stop the flow state. You don't ask questions about it, because if you do, it takes a while to get back. So I keep talking about this. So anyways, first thing that we did was like, okay, we're trying to make and trying to get a sort of build a hypothesis, like why is it more stable. So the way of thinking here that you expect is, what is the most important thing for an organism to survive? if you know anything about biology or biotechnology, you would answer the genome, right? The genome is what is the like central thing to an organism. So why would an organism's DNA be more heat stable than just a normal, like a mesophilic one? What could it have that makes it more heat stable? So if you're aware of the biology of the DNA, there are four nitrogen bases and there are two kinds of bonds, right? Not exactly two kinds of bonds, but two different number of hydrogen bonds. So there's adenine and thymine. They have two hydrogen bonds, and uh, like guanine and cytosine, they have three hydrogen bonds. So then, because they have three hydrogen bonds, it makes them more heat stable. So it's very natural to think that maybe like thermophile organisms, the thermophile bacteria have more GC in their DNA, which makes the DNA more stable. That is a very natural line of questioning to follow. So, in order to do that, you would have to first explore the DNA, right? Because now you're saying that, okay, you're just claiming that maybe because it has more GC, it is surviving. So, I'll show, let's just go to the dashboard. Yeah. So, here's the dashboard. Okay. Like, give it some time, it's going to load up. Yeah. So you need evidence first of all, right? So what I did was I fetched CDS FASTA files. So if you're aware of biotechnology, you know what FASTA files are. They're basically sequence files. It could be nucleotide, it could be protein, DNA sequences, RNA sequences. But I downloaded CDS FASTA. That's you learn new things when you are it's like, I think it was Viktor Frankl in his book, A Man's Search for Meaning. Like, you give a man a why, he'll figure out the how. So, because of a quest, because of an investigation, because you are sort of cosplaying as a biotechnology or a biology Sherlock, you will want to find the how, right? So, you learn a lot of things along the way, which a textbook will teach you, but it will not be relevant in the order that you would want it to be. I cannot explain that statement, but you have to just read between the lines. So CDS FASTA stands for coding sequences. So when we are just going to see the genes that code for things, or yeah, we're going to obviously see the CDS regions, like coding regions. We don't care about the other ones, right? That is why we download the CDS FASTA files of Thermophilus. But whenever you are trying to establish a difference in something, you need to have a control. That's basic science, right? So what are we going to use as our control? We need a mesophilic organism that is like, that thrives in normal environments. And what could be better than our very good friend E. coli, right? E. coli is like this stable organism that biotechnologists, biologists, even biochemists or geneticists like to talk about. Geneticists, I would not say E. coli, but maybe they like to talk about. I've seen they like to talk about CRISPR an awful lot, and it is worthy of talking. It's an amazing technology. So, why are we taking E. coli? Because we need a frame of reference because E. coli is a very well studied mesophile. So, it's the vanilla of genomics, I would say, like standard mesophile. So, that's our control, and we're going to plot. First of all, what we're going to do, we're going to see the GC percentage, uh, like the G's and C's of the genetic material of those both organisms. We're going to plot like the number of genes, the percentage of GC content, as you can see over here. We'll dive into the code later because that's sort of the boring part. Uh, we'll do the interactive stuff right now. So if you can see, it's very well spread throughout. Like it even has genes that have 20% GC content. Most, like, let's see, not, I would not say most, but like around 30-40% of them are below 50% the genes. So because more GC equals more thermal stability, you can see right here most of the genes, like it starts over here. Even let's say the minimum level of GC content in a thermophilus gene is around 50%. So that is telling you something like, okay, like it has more GC, it has a GC bias. So now we have sort of validated our claim that because of more GC, it is more heat stable. So we could stop here, but science is not that simple. Now that we've seen it has more GC, so we have this natural question that now what? Like, If it has more GC, which are the one, which genes are the ones that have the like most GC percentage. So as you can see, I went and saw, because we are trying to see now, we validated like because of DNA, but now we're also, we cannot just say because the DNA has more GC, that is why it's heat stable. That would just be saying like you know our fridge cools because it's connected to the electricity. No, no, no. That's such a weird analogy. But like, it's like saying plastic burns. But we're not seeing why it burns, or is it really better than wood as a burn. Obviously, plastic is not. So now we need to validate further, because uh, just DNA is not going to make something heat stable. It also has to have internal functional functions that make it heat stable. And what drives internal functions? Proteins. So now we're going to see if those GC rich genes are coding any proteins that are helping, like the organism survive. So what we did was took the top 10 genes, GC rich genes, and then got their protein ID from the header of the file. Basically, the file, when you download CDS files, it has. I wish I could show you. I think I have it in my notebook. Like here, it is visible. I know it won't be, anyway, but okay, I'll just show it here. So if you can see, this is sort of the gene ID. In CDS files, each gene is sort of annotated, like with not annotated, but like each gene has a starting header. So you can see each gene has an ID. So in that we have this thing, the protein ID. So we can use the protein ID and access it via the enters API and get the function. So that is what my program did. Now here's something called CAI. We'll talk about that later. But yeah, so you can see the first one is a helix turn domain containing protein. Then we have chlorides and a universal stress protein. So if you can see, it has a helix turn like protein. Then I went on like NCBI and searched for this protein ID, and turns out it's like a SOXR family transcriptor regulator gene. Basically, it's like central to a stress response. It's trying to, maybe the organism uses it to, uh, sort of sense environmental stress. So these are like sort of control genes that help it stay stable under pressure. So we can do that for every single one of these, but I did not. But you can see like the most of them are like transcription factors, DNA binding proteins, heat stress related domains, like this one, helix turn domain. So based on that, we can sort of uh, infer that the GC rich genes, because of the environmental pressure, you could say, uh, are the ones that are like central to its function. Because without these genes, I don't think it would be heat stable. I mean, it's not like I don't think. Obviously, these are the ones that are like central to its thermal stability. So I did not do this just for Thermophilus. I also did it for E. coli. So E. coli, this genes, if you see here, I took the G series genes over here as well to see like uh, what genes are stable, like are the ones like the necessary for function and all that. So it has regulatory proteins and enzymes. So here I'm starting to build a sort of understanding that GC rich genes are the ones that are vital, because GC rich genes are more stable because they have more GC in them, right? So this is a sort of side hypothesis or side inference that I'm making, that okay, very important ones are usually protected, or rather favored with GC richness, so that they survive better, so that the organism also survives. So then we moved on. So now we've taken the DNA, we've taken the genes. Time to dive further deeper in. So then we dive into the codon preferences, codon bias. So we humans, we speak same words, but our accents are different. Someone could talk like, "Oi mate, all this American stink we all sound like this." Or someone might talk like this: "I don't know what you're talking about. It is quite Russian." I don't know what accent is this. And then there is this normal stereotypical Indian accent that people think that Indians speak, but they don't. This is how normal Indian speaks like. Is this Harold and Kumar and all those series are stereotyped. But I sort of even I like that. Okay, it's fun. The telecaller voice is a staple and I like it. Okay, it's funny as long as it's not being racist. So the I keep forgetting that this is an academic endeavor and I'm creating this for the purpose of learning. Keep making jokes. So yeah. So because our accents are different, just codons also have an accent. That is something I did not know until I sort of went through this. Maybe they taught it in my lectures, but I slept during my lectures. Plug into my LinkedIn series. If you are not following me on LinkedIn, go and follow me on LinkedIn. Deep Dalvi. I create content. I have a series called I Slept During My Lectures where I explain biotechnology concepts in a very simple way because I slept during my lectures. I did not, but I was like there but not there. It's like Doctor Strange in his astral form kind of thing. So coming back to the accents, codons have different accents. Like you know that codons are redundant. I keep assuming audience knowledge base. That is the last thing you should do when you're teaching. Yeah. So codons, the like the triplets of RNA, the one responsible for proteins, like uh, making proteins, serve as blueprint for making proteins. They have like they're redundant. Like there are multiple codons that code for the same uh, amino acid. So even uh, cells or organisms have preferred codons. Like for example, there are four different sellers of potatoes, but I only buy my potato from Mr. X because I just like the way his potatoes are. So even like that, I have a preference for a seller. Uh, Organisms have a preference for a codon. Like they will just take GCC because well they just like GCC. Even if GAG is doing the same job. This is just an example. It's not, but like or it I don't know. Like even if the same different codons are doing the same job, that's if the cell prefers that codon, it's going to choose that. Not exactly choose that, but like we'll get to it. So why do codons, why is there a codon accent? Like why can't they just have everything? So it depends on a lot of different factors. Like our cell is basically sort of a factory, and factories want speed and efficiency. And there are multiple things like tRNA. Like there's something called a tRNA pool. tRNA is like basically what assembles the protein on the basis of the codons. It's sort of like the guy that is standing on the conveyor belt in a food factory video. Like he's the one that's taking the bottle and packing it in. It's that kind of guy. So the tRNA is sort of taking the, reading the codon's recipe and it's like placing the stuff. But then the tRNAs are also sort of specific to the recipe. I mean, you cannot give an Indian recipe to an Italian chef. He's going to surely fuck it up. And vice versa is also true. So, like organisms evolve in a way that the preferred codons, like the codons that they prefer, match their most abundant tRNAs. If my city likes Gujarati food, like sorry, that's very niche. If my country likes Indian food, we're going to have Indian chefs. Most of the chefs are going to be Indian. It's like that sort of thing. But then you give my country an Italian recipe, it's going to take time. With the same analogy, if the codon is not preferred, it's not like the cell will not make the protein, but it will take time because it does not have that specific tRNA in abundance. So then what will happen? It's going to take time. So that is, like that is minus, that's a price cut on price cut. That is a cut on efficiency, and we don't want that. So uh, that is why cells have a preference. So when I counted codons in Thermophilus, it relied heavily on the codons that ended in like GC, like they had GC. Whereas E. coli is like a chill guy, he doesn't care. Like GC, AT, GT, GT, whatever. Just give me codons, man. I'll have it because it is a mesophile. It you know mesophile doesn't really sort of it doesn't care like okay give me GC rich, but because Thermophilus needs GC in its let's say genome, it is sort of, that is why it has more GCs, right? Because we are looking at it like inside out. Like if we try to look at it like this, because its DNA has more GC, its codons also have more GC. Therefore, the tRNAs are also more GC. So it is sort of, like a self-supporting kind of thing, that it prefers GC-based codons. That is what we can see, because most of its codons are GC rich. But when you look at E. coli, it really doesn't have sort of a preference. I mean, yeah, it does have most like CTG, but it's not as strong as Thermophilus, right? It's relaxed and balanced. So uh, now that we have, like a, again, now we've dived deep into codon usage as well. So now we know that Thermophilus prefers GC ending codons. So now, uh, again, we should stop here, but again, it's not that simple. So now we'd make a heat map. I hate heat maps. Personally, I can infer a bar chart just by seeing at it. I can infer a scatter plot by just looking at it. But heat maps, I have, like, it takes me time to understand a heat map. Maybe it's just my math averseness that comes in here, but yeah, it's a personal rant. So, normalized codon bias, thermophil. So now, what is normalization? I was, see, like I said, it's very hard to explain stuff, but if you are still watching this, I would very well love to explain. Or if you're just doing a 10 second plus, if you stumble on this, it's going to help you. So normalization is like a mathematical operation, I would say. uh, but in our case, normalization is basically, here we've counted codons in the rawest form, like, okay, but some amino acids like leucine, like I said earlier, that leucine has 6 codons, but some other amino acid only has 1 or 2 codons. So then it's not really fair to count leucine as a more used codon, because leucine just has 6 codons coding for it, whereas some other amino acid doesn't really. So that's not a fair comparison to draw. So in order to, like, draw a fair comparison, we do something called normalization. So instead of asking, how often is GGC used, we ask, of all the codons that like code for glycine, how often it is GGC. So we are comparing preference, not just volume. We are looking for specific ones. So now we have these codon accent maps. Like we here we can see that it is leaning heavily on GC rich codons. Like here, you can see the color coding and all. See, these are all relative, based on relative codon for like preference. They're not really preferred as well. But GC ones, they are. You can see here like ATG, the TCG, and they're all preferred a lot. So there's that. So we have sort of, like stumbled upon the dialect of Thermophilus. It only speaks in GC richness. So like it has a codon bias towards the GC richness. So we even drawed a fair comparison, and now we 100% know that it has a GC bias. What do we do next? We do the same thing for our control. As you can see over here, the codon preference is, you can see there's a lot here that, are preferred ones, like their the genome matches the organism's codon preference. That is a thing. uh, I already explained it, info from that. But as you can see here, it's very well balanced. I mean, there's no uh, rigid codon preferences. Like there are, but like, chill guy is a chill guy. Whereas here you can see like it falls, or like a lot of codons just are preferred more. So it's like a skewed bias. Um, so now, again, what do we do next? We do something called CAI. So what is CAI? So I was going to explain the entire code, but then I realized that it would be very, very, very tedious and then boring, and then people would sort of jump out. And the reason I'm making this video is to explain my, like, it's not even a project. Explain my endeavor, or explain my quest. So it's okay if I just jump over a few things. But then CAI normalization, all these, I thought they were like very essential. So if there's any other questions, you're free to put them in the comment section. So what is CAI? So far we've seen what codons are being used and how, like preferences, but about how well are they being used? How are we going to see that? So that's why CAI comes in. CAI stands for Codon Adaptation Index. It's like a 0 to 1%, 0 to 1. What? There's a score between 0 to 1, like just like here. That says how well a gene's codons match a favorite one. Like I said over here, that these are the ones being preferred, like for methionine. Obviously, this M is methionine, right? Yeah. So these are the ones that are being preferred. So uh, like in CAI, what we're trying to find is like we're trying to see if they're optimized, if genes are optimized. Like for example, uh, if there is a gene that is really important to the cell, like something like a heat shock protein, the cell would want the gene to be read very fast, very and translated like very efficiently without any kind of stalling. So it should have codons that the cell is best equipped to translate. Like I said, the tRNA pool and that entire story, it applies here as well. So when there's a high CAI, you can see like most of the genes in Thermophilus are very well optimized. They match the organism's preference. So when there's a high CAI more than zero point 0.9, it just means that the genome has preferred codons, it is highly expressed, and evolution has really invested in you. It shows that. So in order to make this comparison, what I did was I built a fake reference set. There is something called a CAI, like it if you take known genes, and there's an entire different process for it. But for this kind of comparison, it's a fake reference set is enough. So I just assume that the most used codons in the genome are the ones that are preferred. Like if a guy keeps eating burger for dinner, like uh, 22 days out of the 30 days of the month, I think it is safe to say that he likes burgers, right? So so then we plot the scores, and then we get like most of the thermophilus genes, like they're more than 7 point like 0.7. So they are efficiently translated, therefore they're tuned for survival at higher temperatures. Whereas if you look at E. coli, I mean, I mean, this sort of doesn't match. It does. They are optimized, but like they are for metabolic efficiency rather than resilience. You can see there are certain genes that are on the low end as well in E. coli, whereas Thermophilus has a very tight set of genes. So now, what do we do? Then we do a CAI versus GC plot, GC percentage plot. So I just did this for like sort of further investigation into if GC richness really does mean uh, having efficient, or having preferred codons. So if you can see, higher GC content doesn't necessarily mean that it is like it has a good CAI. And even in E. coli, if you look at that, a higher GC doesn't necessarily mean good CAI. So CAI and GC percentage are can be seen together, but they are not really co-evolving parameters in a way. Like they don't improve together necessarily. So for that, for further, uh, you know, like for further exploration, I did like for the top 100 codon optimized genes. So like most of them are going to have a good CI. So that is why I plotted them on GC percentage. Again, we're getting that it's not necessary to be GC rich and be, this is for Thermophilus and this is for E. coli. You can see it's not necessary that they have a higher GC content and CI. Maybe this is a dumb comparison to make, but then like I said, I was just playing around. So this is sort of correlations. You make dumb correlations, but then you get attuned to making correlations, you get attuned to plotting data, then you get to know what metrics you can use. So then maybe when a real life situation comes or a real life problem comes, you will be very well equipped to draw conclusions from data, or even uh, design the kind of experiment that you want to do and what kind of metrics you're going to use for it. That is why fooling around is pretty important, right? I suggest you fool around with a quest like this, or rather, I would say, do this. Show me your outputs. Do better than me. The bar is very low. It's in hell. So um, I do that. So basically, for like I'll do a quick recap of sorts. So first of all, we compared GC percentages. Because we were trying to find out if GC richness is what makes it more stable, because GC richness would mean a stable DNA. So we got to know that, okay, yeah, Thermophilus does have a higher GC percentage. It has a higher GC content. So that, okay, that is likely to, like, have better thermal stability for the DNA. And then we try to see if those higher GC rich genes, like top 10 genes, they are really even crucial to the function. So that would mean that they are the ones that are important to the survival. That's why they've been sort of favored by GC richness to, like, survive. So you can see there were like this is like a SOXR family stress related protein, so that it helps detection of stress of sorts. So these are like transcription factors, DNA binding proteins. So because they're GC rich, it sort of suggests that they are important for thermal stability, because they also serve the functions for better thermal stability. And then we draw the same conclusion, not conclusion, we draw, like, we try to make inferences by the control, which is E. coli. That, okay, E. coli also has like GC rich genes, but they are enzymes and regulatory proteins. And they are also vital for cellular function. Uh, but this is for metabolic and regulatory needs, but not for heat stress. Then we got to, but then because now we know, okay, DNA is stable due to GC richness, then we dived into codons. We got to see that, okay, Thermophilus prefers GC rich codons, GC ending codons, whereas E. coli is pretty balanced and does not have sort of a bias. Then each organism has a unique codon accent. So Thermophilus prefers GC ending codons. Uh, so because mRNA stability and protein translation efficiency. So this is like a raw thing that we did. Like we try to plot codon bias, that which one is preferred. So we got to know that a lot of codons are GC ending in case of Thermophilus, and they have, like, the relative codon preferences high for them. But in the case of E. coli, really doesn't have sort of codon preference. It's like balanced of sorts. Then we went to CI. CI is basically trying to see if a codons, like if an organism's most used codons uh, are like aligned with the genes that, genetic codons that it has. Like if the genome is really having codons that are preferred by the organism. So how would we got to know that? I made a fake reference set by just assuming that most used codons are the ones that the organism prefers, and that's like a safe assumption to make. Then we got to know that most of the genes in Thermus thermophilus have a more than 0.7 more than 0.7 CI, like they're optimized for efficient translation. Then in E. coli, it has optimization, but for metabolic efficiency, but the distribution is broader and less extreme, whereas here it's very extreme. Then we plotted CI versus GC. I just wanted to see if like GC richness also so correlates to, like, codon optimization. And if yes, like that means like the genes just aren't stable, but they're also priority builds in the genome. Like if there are the genes that are also codon optimized and GC rich, that is like a alternative assumption to make. Before, when I was talking about it, I told that it's not necessary that CI and GC are co-evolved parameters, like they can be sort of separate. But if a gene is being preferred by both, like GC content, is there was something over here like GC content, like, as you can see over here. This is in case of E. coli, but like I'm just trying to explain a point here. If it is being preferred in case of GC content and in the case of codon adaptation, it is like, a natural connection to make that it's pretty important for the cell. This in case of E. coli, it's a xylonate dehydratase. Maybe it has something, like in the metabolic pathways involved in that. In our case, we have this one. See, this is the most GC rich gene in case of. So that is the safe assumption. So when you try to play around with the data, you get to learn inferences like these. You obviously have to validate them. If because if you're wrong, you will learn the wrong thing. There is the danger in autodidactism like this. But because of AI and all that, these dangers have been minimized as long as you have like better, good search fluency and a good feedback loop with yourself. Like sort of that sense that something is wrong. And how do you build that sense? Question yourself at every single step. So then I wanted to see like this is the same thing, but like in case of codon optimized genes. So most of them are going to be here, but I wanted to see the GC percentage, like the top GC rich genes. So where do they stand? Like trying to see the same inference from here, but in case of more genes, so that I have like I can see it better. So as you can see over here, there's like it has a higher GC content and also codon optimized. But as you can see here, there are certain ones that are not codon optimized. But in case of Thermus thermophilus, like it has a very tight gene set that is codon optimized and higher in GC content. But as you can see here, it starts from 58 and ends at 67. Whereas here, the starting GC content is like at something around 74.2 or something. So it's already GC rich and it has a core gene set. This is basically if you were to uh, draw parallels, this is sort of the same thing from the first graph, sort of. If you can make the connection, you know. So now at the end, like TLDR, Thermophilus has GC heavy genes for thermal stability because GC just makes the DNA more stable. It has GC ending codons as its accent, as its preferred codons. It has a high CAI because the genes are optimized to be translated efficiently without any kind of stalling. So therefore, they are like, the stress resilient genes have a high CAI. And then it's not just surviving the heat; it is optimized by years of evolution to survive the heat. Like it's like it's that one meme. Like I'm built for this. I was born for this. Like it's that kind of thing. So what did I learn from all of this? So I learned how to parse FASTA files with BioPython, how to compute GC percentage codon frequencies, CAI. What is CAI? I was not taught this in my syllabus. What is a codon bias? How it reflects adaptation. There are even like beginner level tools like these. If you are like brave enough or let's say hardworking enough, you can reveal biological strategies of nature. So, but the most important thing here is that I learned bioinformatics. Like this is an exercise sort of in comparative genomics, all by myself. I have not validated it with any kind of professor or something. I should have, but that is like autodidactism is like that. You learn by yourself. You learn like this. You keep stumbling, and then you suddenly build a rocket. So there's that. So it feels like I'm trying to solve puzzles, and it's fun this way, like a detective. So thanks for joining on the first episode of Data Disco. And next time, who knows? Like, but I've already told you what is there next time. But like next time, I might switch up uh, antibiotic resistance, ancient genomes, or maybe we're going to design a gene of ourselves in silico. Uh, so stay tuned. This project was powered by Python, driven by caffeine and my ADHD fueled uh, dopamine seeking flow state, and it was guided by curiosity. Uh, if you found this helpful, share it. Maybe I'm always open for a talk. Comment below, or engage with me on LinkedIn. Follow me on LinkedIn. Deep Dalvi, that's the name on LinkedIn. And if you want to build your own quest, hit me up. But yeah, but before that, let's go through the like code as well. So and this is my email, by the way, if you want to email me. So you can see over here, like we take the codons, we count them, build CI reference, we're creating the fake reference set over here. I will soon put this out. Stay tuned, keep watching the description because I have to clean this file. It's like about 400 lines almost, but more like a lot of it is also because of the dashboard thing. I cannot stress enough. I have seen people with smart, really good, like inferences to make, not know how to code. I mean, it's okay. I don't want to be that guy because I know what the tech market is right now and learn how to code. But for bioinformatics, you gotta learn how to code. I mean, obviously, that's like the minimum requirement for it. But just don't learn how to solve problems of computational biology, but rather learn visualization as well. Because I'll show you. This is my dashboard. Okay, this is the one, right, that I showed you. Fine, right? Now, when you spend more time on it, when you try and optimize it, try to beautify it, try to make it look better, I'll show you what happens. You can create very good looking things, and because uh, if things look good, they're more to be click more likely to be clicked on and like that. So just don't learn how to solve computational biology problems, but rather try to present them in a good way. Because if you know even the unified theory of physics, but if you cannot put it into words, it's of no use to anyone. So just like that, if you've solved a very good computational biology problem, but you are not able to represent it in a very nice way, it's of no use. So I'm, I cannot stress on this enough, but like learn how to use UI elements, learn how to use themes, learn how to use, like it doesn't now. AI has made it so much easier to learn and even like code. I would say that. So yeah, before I'll have to get out of this. What? Why is it not running? Oh yeah, I'll have to first. I'll have to first stop this one because they are all both running on the same thing. So there's that. So yeah, while we wait for that to happen. So now if you can see, this one is almost the same, but like this has more colors, more funkiness to it. Now side by side, like comparisons, more popping out UI, not even UI, we're not using anything, but like looks good. Presentability. That's at least that's what I think. So pretty soon I might, put it on GitHub. I will have to anyways. So this was the first episode of Data Disco. I hope you liked it. And if you stayed this far, genuinely you have the patience in me to hear me blabber and you're willing to learn. That shows. Hit me up uh, uh, on my email because if you made it this far, you must have seen my email. Send me an email saying Thermophilus enigmatis, and I will personally reach out to you and I will show you some of the different strategies, secret ones that I use that I'm not yet ready to put out in the world because I'm working on them, but maybe you can be a part of it. So, see you, and I hope you learned something here or at least had fun. I don't know. Learn something, had fun, both of them at the same time. Very good for me. So, with that being said, bye bye. Take care. Have a good day. Have a good night. Have a cup of coffee. Have a cup of tea. Whatever. Just have a good one.